Okay, in this video, we'll go over Chapter 20, the urinary system. Okay, part of keeping a stable environment or homeostasis is to maintain the composition, the volume of various body fluids and pH within a fairly narrow range of normal limits. And the whole point of the urinary system is to remove metabolic waste, such as drugs and toxins that your body doesn't really need. And the whole system is really made up of 2 kidneys, 2 ureters, the urinary bladder, and the urethra. Okay, here's the generic image of the urinary system. Of course, the kidneys here, with the adrenals sitting on top. The ureters going down to the bladder, and then to the urethra. Alright, we'll talk about the kidneys first and kind of work our way out of the system. Kidneys are a bean-shaped organ, reddish-brown in color. In the adult, it's about 12 centimeters long, about 6 centimeters wide. And it's wrapped in a very fibrous, very tough a membrane called the capsule. This will help maintain that kind of bean shape to it. Alright, the kidneys lie on either side of the vertebral column, and it's actually behind the abdominal wall. And this is described as being retroperitoneal. Retro for back or behind, and they're actually behind the parietal peritoneum. And this actually will be shown if someone has kidney stones. It may appear to the person as having lower back pain, as opposed to actually being something wrong with the kidneys. So the kidneys aren't in the abdomen; they are behind that abdominal, that peritoneum cavity. The left kidney is about 2 centimeters higher than a right kidney, because the liver is kind of squished down on the right side, on the right liver or right kidney. See the size or the height difference here. That one's a little bit higher than this one, because you have the liver sitting right above it here, kind of squishes it down. The kidney has 2 very distinctly different regions. These are terms that we've already talked about before, and we'll mention in future chapters also. Renal cortex and renal medulla. Of course, renal is always a reference to the kidney. Cortex is always the outside of an organ or structure, and medulla more toward the middle or the inner part. Alright, the portions of the cortex will project inward toward the center. And these are called renal columns. They actually look like columns on a building. So even though they go into the medulla, they are still projections of the cortex. And the shapes that these columns form within the medulla are called renal pyramids. They actually look like pyramid-shaped features. And at the very tips of these pyramids are a structure that looks kind of like a cup, and these are called minor calyces. And calyces is plural. Singular would be calyx. So you have multiple minor calyx and they're called minor calyces. Okay. Of course, a drawing of one. Here's a real one. Cortex here. Then the medulla in here. And these projections that go down here and here. And here are the columns. And the triangular shaped basin looking things, these are the pyramids. Here, 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 here. At the very tips of these are the minor calyces here and here. On the real one, you actually see the difference in color. The lighter color would be the cortex, darker would be the medulla. That's a pyramid, that's a pyramid, that's a pyramid here. And the very tip would be the calyx. Alright, from there you have multiple minor calyces forming, or actually merging together, forming a major calyx, or major calyces. And these major calyces will merge with each other to form what's called the renal pelvis. This is a basin in the middle of the kidney. It's almost a funnel shape. This will help drain the urine from all the pyramids that's been collected. And actually the pelvis is actually the very top part, the superior end of the ureter. So the function of all these calyces is to drain a little bit of urine. So you get a little bit of urine from the minor calyces, all that combined form major calyces, so you get even more urine. All those dump into the renal pelvis, and then the pelvis will form the superior end of the ureter here. So the pyramid, at very tips of those would be the minor calyces. They merge together to form the major calyces here, and this whole thing is the pelvis. So all the urine will kind of collect here and then dump into the ureter. And from here you'll go into the bladder. Again, this is a term we've talked about many times before. The space where you have vessels and nerves and the ureter and lymphatic vessels enter the kidneys is through the hilum. Remember, the hilum is not particular for any organ. We talked about this for the lymph nodes and the spleen, the lungs and the kidneys. There's any indented area where you have vessels or nerves entering a structure. And from the hilum, it will enter a larger hollow chamber called the renal sinus. And this is basically everything in the kidney itself. This is where you'll find the pelvis, all the calyces, the blood vessels, the nerves, and the fat. So everything found inside a kidney is in the renal sinus. All right, we'll talk about the blood vessels. The renal artery is the vessel that will enter each kidney and will branch smaller and smaller and smaller until you get down to the functional unit of the kidney called the nephron. This is where all the work gets done. And the final branches of this renal artery that lead into the nephron are called the afferent arterioles. Now, we've seen this term before, afferent, when we talked about the lymph node. Both afferent and efferent are generic terms that mean something very, very particular, and it's very important to keep the two straight. Afferent structures always arrive at a structure. So afferent for arrive, then efferent are always exiting a structure. Alright, here's how the inside of a kidney looks when it's cast in plastic. All these are leading to various nephrons. Of course, the red would be the arterioles, and the blue would be the veins going back to the renal vein here. This dark purple one here, that would be the renal artery here. And this yellow thing would be the ureter, which would travel on to the bladder. All right, in each kidney, you have roughly 1 million nephrons, so you have about 2 million total. And the nephron is composed of two parts, the renal corpuscle and the renal tubule. Now, it's basically from here on that the terms and the processes get fairly complex, so you need to keep all these terms straight. All right, the first one, the renal corpuscle, is the filtering unit made up of a very tangled mass of capillaries. That's called the glomerulus. And surrounding that glomerulus is basically a cup or a sac-like structure that collects everything that gets filtered. That's called the Bowman's capsule. So the corpuscle is the glomerulus, which is a tangled big wad of capillaries, and plus the Bowman's capsule. Both of those together make up the corpuscle. All right. Like we mentioned before, fluid will enter the glomerulus through the afferent arteriole. Will get filtered at the glomerulus. And then the fluid will exit the glomerulus through the efferent arterioles. So arriving at the glomerulus through the afferent, exiting the structure through the efferent arteriole. And this is the very first step in the formation of urine. The actual glomerular filtration, what this is. All right, here's how one actually looks. One big tangled mass of capillaries, and the renal tubules kind of scattered all around it. As how one looks on a slide. A big tangled mass glomerulus here, and renal tubules here. Again, this is a real image of one on a real slide. Alright, and like I mentioned before, here's where the terms get fairly involved, but if you break down the names of the term you're looking at, it tells you where you should be looking. And I'll put up an image that has all this together, and I'll probably go back and forth from the text and then to the image. Alright, the renal tubule, once it leaves that Bowman's capsule, it becomes very, very twisted, very convoluted. So this very first section of the tubule is called the proximal convoluted tubule. It's called convoluted because it's very twisty and windy, kind of bent all over itself, and proximal because it's close to the Bowman's capsule. Alright, from there the tubule will continue into the medulla, leading to what's called the loop of Henle. As a loop because it has a part that goes down, or the descending limb, and the part that goes back up, or the ascending limb. And both of these together make up the loop of Henle. And from there the tubule will head toward the Bowman's capsule again. It's still fairly twisted, so it's still convoluted, but it's now distal from the capsule. So it's a distal convoluted tubule. And these distal convoluted tubules will merge together from other nephrons and form what's called the collecting duct. As these ducts that will continue through the medulla to join other collecting ducts that will drain urine into the minor calyx. And we'll start with the generic kidney, and we'll get closer and closer, and we'll talk about the terms we just mentioned. So if you're zooming in, say on this area here, the cortex and the medulla, and one nephron. Okay. This image you'll see quite often, especially in lab. And here's how you need to look at this: is follow the arrows. So we'll start here with the blood entering the glomerulus. All this looks kind of like a crown, would be the glomerulus. So the blood entering here, afferent arteriole, it gets filtered through all these capillaries, then will leave the glomerulus to the efferent arteriole. Then the blood will continue on through arterioles, the capillaries, venules, veins, and then back to the heart. The stuff that gets collected is caught by the Bowman's capsule, this sac-like structure around the glomerulus. And at that point, the stuff is called filtrate because it's been filtered. And the stuff that's in yellow here is the renal tubule. So this first portion, very twisty, very windy, the proximal convoluted tubule because it's close to this capsule here. Alright, from here, you'll have a part that goes down and goes back up. So this is the descending limb, and that's the ascending limb. But both of these together is a loop of Henle. And listed here as the nephron loop, but it's the same thing. So loop of Henle. I'll talk about this in a little more detail here in a second. Then from there, the tubule will go back up. It still gets kind of twisty and windy, so it's still convoluted, but it's now distal from the glomerulus. Then we'll go into the collecting duct here. And then we'll go on to the minor calyx, and then major calyx, then to the renal pelvis, and then to the ureter, and then to the bladder. So this is just one nephron of the approximately 1 million in each kidney. So you have about 2 million of these. This is where everything gets filtered within your kidneys. So capsule here, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and then the collecting duct. Again, the red for the arteries. Entering the glomerulus here, afferent, A4 arrive, gets filtered. Stuff that can't get filtered remains in the blood and will leave the capsule to the efferent arteriole, E for exit. Now there are two different types of nephrons depending on how close they are to the medulla and how long their loop of Henle is. The first one, the cortical nephron, is found mostly in the cortex. That's why it's called cortical. It's fairly far from the medulla, and these have shorter loops of Henle. And of course, the opposite of that would be juxtamedullary nephron. These are very close to medulla and have very long loops of Henle. So here are those side by side. Here's the Bowman's capsule here, mostly in the cortex. That's why it's cortical, and a relatively short loop of Henle here. And then, if this is the border dividing the cortex and medulla here, This one's very close to that border, so it's going to be juxtamedullary and a much longer loop of Henle. So make sure you keep those two terms straight. Alright, now we'll move on to the formation of urine. Of course, the main function of nephrons is to control the composition of body fluids and remove waste from the body. And that product is then called urine. And within urine, you'll find, of course, water, waste, electrolytes, and so on. And urine is made by 3 separate processes. And one we've already talked about before. When the stuff gets filtered at the glomerulus, glomerular filtration. And then you have tubular reabsorption, and then finally tubular secretion. These last two terms, tubular reabsorption and tubular secretion, are very confusing based on their name. When we go over what happens on each one, it sounds like the names should be backwards. And I'll tell you a good way to keep those straight. But pay attention to these two, reabsorption and secretion. The name almost makes it sound like what you're looking at is backwards, but almost always confused with each other. It's very easy to get those confused. A recap of the glomerular filtration. You have water and small dissolved molecules can filter through the glomerulus, those capillaries, but some can't because they're just too big. Think of the glomerulus as like a pasta strainer or a sieve. When you're making pasta and you pour it into the strainer, water can get through, but the bigger parts of the pasta can't because they're just too big. And stuff that is too big will continue on into the blood. And stuff that does get filtered goes on through the renal tubule and continues on the process. Now the glomerular capillaries are going to be a lot more permeable than regular blood capillaries because they have additional holes kind of spread out through the glomerulus. And of course, the filtrate is formed as things are filtered from the glomerulus and then caught by the Bowman's capsule. And here's how this looks. The big tangled mass of capillaries, the glomerulus, Bowman's capsule, ready to catch everything that gets filtered here. And a lot more holes kind of spotted throughout the glomerular capillaries. They're called fenestrae. Alright, the main force that will move fluid within the glomerulus is called hydrostatic pressure of the blood. And this is done by something actually called Bernoulli's principle in physics. The efferent arteriole is actually smaller in diameter than the afferent arteriole. And so this difference in size will cause blood to move faster as it exits. That's what Bernoulli's principle is. Fluid will move faster in a smaller space. This is also how airplanes get lift. And a good example of a way to think about this is putting your thumb over a garden hose. You have a amount of fluid, but going through a much smaller space, so that when you put your thumb over the hose, the water will shoot out faster. So that's what's happening here in each glomerulus. All because of the size difference between the afferent and the efferent. So efferent is going to be smaller. Okay, so you have fluid coming in to the afferent arteriole, but then efferent is smaller, and that size difference will make blood gets filtered faster. So the faster things are filtered, the faster things can get put in the urine and then get gotten rid of from the body. Let's see, the glomerular filtration rate, or GFR, is going to be directly proportional to how much pressure there is. So if filtration pressure goes up, the amount of filtration rate goes up. So the higher you turn on a water faucet, the more water comes out. The lower you turn down the pressure, the less water will come out. And the measurement of GFR can indicate various things that are going wrong with the kidney. Could be kidney stones, could be enlarged prostate for males. It will indicate some type of renal failure. So there's a range of what normal GFR should be. And when that goes down, then something is going wrong. Alright, tubular reabsorption. This is when substances move from the renal tubules into the blood capillaries. And the capillaries that surround them are called the peritubular capillaries. And this is where most tubular reabsorption will occur, is in the proximal convoluted tubule. So again, tubular reabsorption is going from the renal tubule into the capillaries, not the other way around. This almost sounds like the renal tubule is reabsorbing material, and that's not what's happening. It's the other way around. You have material going from the renal tubule into the blood capillaries. Okay, so here's the blood going through the glomerulus here, being filtered. Tubular reabsorption is going from here into the blood capillaries. Okay. When we talk about water being reabsorbed, this will occur by osmosis. You know, that passive transport that you should have talked about in the first part of the 168 class, or basic biology. Now, other ions and sodium are going to be reabsorbed due to active transport. And even these small changes in tubular reabsorption can mean a large difference in the amount of urine that's excreted. Alright, now for the third part of forming urine, the tubular secretion. This is when substances move from the blood capillaries into the renal tubules. So they're not being secreted by the renal tubules, they're being secreted into the renal tubules. So that's why I mentioned that tubular reabsorption and tubular secretion sound backwards of what they should be, but think of where the material is going. So for tubular secretion, from the blood capillaries into the renal tubules. Now what's going to be secreted is material that the body just doesn't need or just doesn't want. Excess waste, excess water, drugs, and so on. And what's key here is the secretion of hydrogen ions. That will help regulate the pH of body fluids. Okay, and here's both of these last two steps, one graphic here. Renal tubule in yellow, blood capillary up here. Tubular reabsorption from the tubule into the blood capillaries. Tubular secretion from the capillaries being secreted into the renal tubule. And from here on, what's left in here is what will end up in urine and will end up being peed out. So very easy to get these terms backwards. So reabsorption being reabsorbed by the capillaries, and then tubular secretion being secreted back into the renal tubule. Alright, next we'll talk about the regulation of urine, the urine concentration and volume. Now, the ability of kidneys to maintain the stable internal environment really depends on their ability to reabsorb large volumes of water. And the different part of the nephron will have various different functions. And we'll talk about some of the key ones, because this is a lot more complex of a process than we will get into, because we just don't have the time. For the descending limb, this will be very permeable to water, but nothing else. This is where you have a ton of water exiting the nephron through the descending limb. And of course, water will leave by osmosis. It will enter the interstitial fluid. And when you have all this water leaving, what you leave behind in the tubule is a very concentrated solute. So as water, if they have water plus all material through here, as water exits, you have nothing left but solid material. So very, very concentrated solute here. That's only for the descending limb. You basically have water exiting the tubule. Alright, for the ascending limb, it's not permeable to water, but this is where you have ions that are pumped out by active transport. This will leave behind a very weakly concentrated solute. So when you go back to this image, here it's very highly concentrated, but then you get some stuff that's pumped out. What the body wants, what's left over, is what will be peed out. So when you get to here, you have very weakly concentrated solute. And something that's fairly complex that we will just mention by definition, the difference in fluid movement, both on the descending limb and ascending limb, is called the countercurrent mechanism. The amount of stuff that gets pumped out here influences how much water gets pumped out here. So they affect each other, but at different rates. So this is called the countercurrent mechanism. And of course, there's a lot more to it than this, but just know that what that term is a reference to. Here are some numbers. We talk about concentration. Let's say you start off at 300, whatever unit this is in. I don't remember what it is. When you have a ton of water leaving, you get more and more concentrated. So here, we're at 1200. At this point, at the ascending limb, you get material being pumped out through active transport. So you end up with a very weakly concentrated solute. So you start up at 300, peaked up at 1200, and you're left with 100. So a great majority of what you drink or what you fluid that you get from food that you eat will be reabsorbed by the body. You pee out very little, but no percentage-wise. You're only peeing out about 10% of the water that your body takes in. The distal convoluted tubules, these are going to be controlled by hormonal actions like anti-diuretic hormone, aldosterone. This is where water and electrolytes are going to be reabsorbed by the tubule. And the collecting ducts are also controlled by hormonal actions. This is where active transport will help pump out material that the body wants and leaving behind what it doesn't want. At this point, what's left behind will be composing urine and then will exit the body. So the key thing here is both of these are controlled by hormones. If you take drugs that help make you pee, like diuretics, they will affect these two here. Urea and uric acid, these are somewhat related, but they're not the same. Urea is the byproduct of breaking down amino acid catabolism. And the amount of urine that's found, or urea found in urine, is a reflection of how much protein is in your diet. And then uric acid is the product of nucleic acid metabolism, and in particular the guanines and adenines. Remember, these are 2 of the 4 nucleic acids that you have, along with thymine and cytosine. So urea, amino acid breakdown, and uric acid, nucleic acid metabolism. Alright, the composition of urine. Of course, as you would imagine, 95% or so water. And within the urine, you'll find uric acid, urea, and creatinine. You may have some amino acids and electrolytes. There are some things that should not be in urine, some things that should be in urine. And of course, the volume of what you pee out will depend on your intake and environmental factors. If you drink a lot of water, you're going to pee a lot. If you don't drink a lot of water, you're not going to pee a lot. The ureters, each one about 25 centimeters long, will join the urinary bladder in the pelvic cavity. These run parallel to the vertebrae in the vertebral column. And the innermost lining is what's called the mucus coat, is where you'll find the transitional epithelial cells. These are able to expand and then recoil back to its original size. This is a cross-section of a ureter, the mucus coat here. And these cells are able to expand and then go back to normal. Right, from the ureters, you go on to the bladder. Distensible muscular organ in the pelvic cavity. And within the bladder, you'll find smooth muscle that form the detressor muscle. These will actually, when you force yourself to go pee, this is what you're pushing on. The detressor muscles help you move that fluid forward. And within the bladder, you'll find the internal urethral sphincter. And this surrounds the neck of the bladder, is what will prevent the bladder from opening and leaking out urine. People who are incontinent, this isn't as strong as it should be. Women who have had children, or multiple children in particular, sometimes when they cough or when they sneeze, they may get a little pee come out. That's because this sphincter isn't as strong as it used to be. Right, here's both a front and back view of the bladder for a male. The detressor muscles, all right here. It'll help kind of squeeze the bladder, forcing fluid out through here. And there's the urethral sphincter right here. And from the back side, this is the male because it has the prostate. The urethra is the tube that will convey urine from the bladder and then outside of the body. There's a very big difference in size, or I should say in length, between males and females. In females, it's only about 4 centimeters long. In males, it's about 20 centimeters long. And of course, for males, it has a dual function, both for urination and for ejaculation of semen when we get to reproduction. And we'll talk about that in a couple more chapters. So for males, it has a dual function. Okay, here's both comparing to female and male. Female on top, fairly short urethra, and here's a male with prostate and a much longer urethra, about 4 times that size. Alright, that brings us to the end of the urinary system. Of course, if you have any questions, then please let me know, or post them to the discussion board.